Welcome students to the next session of Understanding Animal Kingdom. Today we'll talk about embryonic development. But before we move on to that, we need to first ask us certain questions from the last slide. What is the evolutionary benefits of symmetry? Did you try to think on that? Why are certain animals symmetric while others are asymmetric? There might be certain advantages of it. Think about it. Then, in what ways does the false segmentation of parasites like tapeworm enhance the life cycle? Because if so many animals are metameric, why does your tapeworm have pseudosegmentation? Then, another aspect is, did you think about the topic of coelom, the body plans, etc., in the context of human body? Did you try to explore about those four organs that we talked about? How and where are they located? Did you try to use the scientific terms to explain them? If not, you still have a chance and do have to learn those. So explore further and reflect deeper. And let's come back to our topic of today. So we have to first understand how does the embryonic development actually happens. We know that in most of the animals, the reproduction is sexual reproduction, wherein sperm and egg is involved. So when a sperm fuses with an egg, in that case, a new cell is formed that is called as a zygote. The single cell is the initial stage which has identity of both the parents and then will further undergo mitotic divisions to form embryonic cells, and then which these cells will organize further. The stages through which these cells undergo, the mitotic division through which these cells undergo, are called as cleavage, and which result in the formation of a 2-cell, 4-cell, 8-cell, and exponentially so forth for the increasing number of cells. So now that way we can have these various kinds of embryo. But few aspects that we need to understand in this particular scenario is that whether these cells have a certain identity or not. So let's talk about that. Say for example, whenever these cells are beginning to divide, there are certain animals in which we see that these cells already have acquired an identity. They already behave in a certain manner and they already know where they have to go and so on and so forth. However, that kind of command has its own limitation. On the other hand, what we see in many of the other animals is that the cells do not have any identity at this point, but they acquire that identity over time, wherever they are present and in which local surroundings they are present. And through that, they acquire their own identity in that kind of environment. Again, each of these methods may have their own advantage and disadvantage, which you can further explore. However, that this exists as two different mechanisms is itself a idea that shows that how things evolved over time. The next aspect, so basically, is that we have to understand what is the manner in which these divisions are happening and what structures are forming over time. So the first thing that we have talked about already is whether these cells have an identity or not. So the question of identity is the first thing. Next is, as they're undergoing through cleavages and they form what is called as a blastula, wherein at the point they begin to acquire particular designations for their cellular structures and form what is called as the germ layers, broadly the ectoderm, the endoderm, and the mesoderm. Now, these particular layers also have their own variations, and also there are animals which may not be having all the three layers, and we'll talk about which these animals are and what exactly is present otherwise if they don't have that one particular layer within them. So that is the next aspect, that is in the case of germ layers. Now, once these germ layers are present and the cells are undergoing movements and they're undergoing transformation, they're forming different structures, one of the key unique feature happening during the embryonic development is that an opening called as the blastopore invaginates and forms what is called as the archenteron or the embryonic gut. And now whether the opening of this archenteron is your mouth or the anus for the future purpose, that is another aspect that and that is utilized for understanding animals. So now whether the anus or the mouth came just like the chicken or the egg story, that is the third aspect of understanding embryonic development. Now let's move ahead with the second aspect that you mentioned, that is germ layer. Now why we're talking about it first, because it is key point to understand about how we classify animals, and then we can further talk about those other 2 points which are actually integrated with each other. So based on germ layers, you can classify animals into diploblastic or triploblastic, where the word is basically coming from the number of layers. So diploblastic basically means they have 2 germ layers, and triploblastic basically means they have 3 germ layers, the ectoderm, endoderm, and mesoderm. However, diploblastic animals don't have a mesoderm, rather they have a jelly-filled mesoderm layer. And what we see is that diploblastic feature is found in those animal groups which are more evolutionary earlier in the geological or evolutionary time scale. However, triploblastic feature is found in those animals which appeared a bit later than them. So in the few earliest invertebrate or non-quadrate animals such as sponge and sea turtle, we do find this diploblastic kind of germ layer formation. However, in the other animals beyond them, we do find triploblastic kind of germ layer formation. So that means these germ layer formation would have had certain advantage, and these structures also evolved over time or changed or transformed over time based on how the environment was and how the needs changed over time. So that is another aspect that you can think about. So these images show you how these layers are arranged and ultimately surround what is called as the central digestive cavity. Now, We talked about the chicken or the egg situation or the anus or the mouse situation for this topic today, whether the mouth came first or the anus came first. Through that, we have two different categories of animals, one is the deuterostomes or protostomes. Here, the word deutero and proto are basically referring to the emergence, whether it came out later or first, and stome basically refers to your mouth. So deuterostome is basically animals wherein the mouth came later, and protostomes are those animals where the mouth came first. In deuterostome, what we see is that the cells do not usually identify with any particular identity or feature altogether at once. They determine their features over time based on where they are present, with what local cues they are having, what local surrounding they are present, what other cells are present around them, and so on and so forth. Such kind of development is called as a regulative development. In contrast, what we see for protostome animals is that we already have certain predetermined functions of certain cells based on early cues which are present inside of these cells, and that way they have mosaic kind of feature, because just like a tile of or tile work of mosaic that you see in any beautiful mosaic artwork, just that in same manner, each cell have already acquired their own identity and they are present in a random conglomerate of what is called as a mosaic formation. So another aspect is that these animals, based on the kind of coelom they have, we can classify them into enterocoelomate and schizocoelomate. That is the point we will discuss in the next slide. And deuterostomes by and large include our echinoderms and most of the chordates, while protostomes include our most of the other non-chordates except your echinoderms. Echinoderms referring to the sea stars and other relative animals to it. And chordates are all those animals which have a notochord present. The next aspect of top understanding is the coelom, the cavity that is present between these layers. So whether the cavity that is present within the organism, is it surrounded entirely by the mesoderm for triploblastic animal, or it is surrounded between or surrounded by both endoderm as well as mesoderm in the case of a triploblastic animal. So basically the terms as such are present only for the triploblastic animals and not suitable for a diploblastic animal, because they usually lack as such a coelom altogether. And in triploblastic animals also, we can also have animals which lack the coelom altogether. And for your reference, there are three worm groups that are shown here. The flatworm, also called as the platyhelminths, the annelids, and then you also have been shown here the nematodes. So your flatworms basically appear like a sheet kind of structure. Annelids have this metameric structures where your earthworm also belong, and then your nematode are also called as your roundworms. What we see is that in the case of flatworms, there is no cavity, so they are called as acoelomate. So we look at the actual sections also, how they look like under the microscope in histological sections. Histological basically mean tissue sections. Then we have eucoelomates, where true coelom is as such seen. That is often found in your case of annelids and other higher vertebrates, as well as other higher non-chordates as well, such as your mollusks, such as your other animals, your echinoderms, etc. as well. This layer, if it is surrounded by both mesoderm and endoderm, in that case we call the term as pseudocoelom. If it is entirely surrounded by just mesoderm, we call it as a true coelom or a eucoelomate animal. So this is the image or histological section of a body of a fasciola, also called as your type of sheepworm, which basically causes your digestive system ailment, and it is spread again through contaminated food and water, and it does not show any particular cavity as such, and that is why it is called as an acoelomate animal. Do look at how this structure looks like, because in the next slide you will be immediately flabbergasted how the structure changes altogether. So now in this situation, you can see the pseudocoelomate animal as seen in the scaris, a common disease causing parasitic worm, which causes ascariasis, a gain of the digestive system. Most of your worms are actually associated with your digestive system ailment. However, they do try to hamper with other aspects or other areas of your body as well. But in this case, what you will see is that there is this layer of muscle which is shown in this deep magenta pink, and there are these internal organs such as uterus and intestine and other aspects which are surrounded by an endodermal layer. And your empty cavity is present, this entire white space is present in between that endoderm as well as your mesoderm all the way here. So this kind of cavity is called as a pseudocoelom or pseudocoelom, and these animals are called as a pseudocoelomate animal. And if you turn around this image, it will look like a clown as well. That's another aspect, but let's come back to the point. So true coelomate animals, annelids, and every other animal beyond that, what we see in this case is that they have the coelom or the cavity which is surrounded on both the sides by mesoderm. For example, if you see this intestine here, the intestine is surrounded by your endodermal layer, but over that you see this another faint mauve color muscle layer, and then this empty cavity is present, and then another layer of this mauve color structure is present, both of which are muscles derived from mesodermal tissues. And that way you can see that the cavity is present between two mesodermal tissues, and that is why this kind of coelom is called as a true coelom, or these animals are called as eucoelomate, and the coelom is also called as eucoelom. Now, within true coelom or eucoelomate also, we can further have two classification, that is whether they have a schizocoelom or whether they have an enterocoelom. Schizocoelom is primarily seen in those non-chordates or in those animals which have development happening in usually in the format of your mosaic kind of development that we've already discussed, or which is called as your protostomic development. And what we see is that in this case, the word schizocoelom is actually coming from split. Schizo basically means split. So when the coelom is coming from the split of mesodermal cell, that is called as schizocoelom, as seen in your annelids, arthropods, and mollusks. So these cells emerge out as split, and then they form these cavity-like structure over time. However, in the case of enterocoelom, the archenteron outpockets, and through that your mesoderm arise, and from which your coelom is formed. That is seen in most of your higher animals. Okay. So let's just recall because so many terms have been discussed. We have talked about coelom. We have talked about how the coelom looks like in different animals. We have talked about the nature or connection of these coelom as well as the developmental stages in the context of deuterostomes and protostomes. And we have also talked about the germ layers and in totality how the embryonic development looks like. Now, you can understand about the questions that we left off with the previous session. That was what structures are formed by ectoderm, endoderm, and mesoderm. I have already given you a clue about mesoderm. Try to explore now about ectoderm and endoderm. How is the development in animals different from that of plants? We have already discussed about animals, but do try to think about how it might be different from that than in plants. Then, what is the appearance of coelom in humans? Which organs are housed where? Again, another question that we left off with the previous aspect. We did talk about tube within tube plan, but now you can further expand it along with this in the context of coelom as well. So meet you in the next session now, where we'll start talking about in more depth about the structure that is present in animals.